Okay, now we are good to go. So before starting today's session, I would like to introduce with my Google Drive folder. I will share this folder with you, and you will find all the necessary software here. Okay, or you could download them from their website directly, and there's no problem. But in case of some tools, um, maybe they will need to submit the serial number or sometimes the product key. So that's why I share all the necessary information here. Uh, Especially today, the software that you will use um, is the ChemDraw. So here is the ChemDraw exe file. Just download. You have to download this file, and you have to install this by clicking on it. Okay, just click on your right button of your mouse and then run as administrator, and then here the PyMol several PyMol version 2.5.2, 2.5.7, or also you can download the PyMol from their website. That will be good for you. And there is the BioVia Discovery Studio and the Avogadro and the others. So to not install any software without giving my instruction, and there are also the simulation softwares available, especially the named. Okay, so just download the ChemDraw from here, and here is the serial. Okay, here is the serial number for ChemDraw. So when you install this ChemDraw exe file, then they will ask you for your serial number. Then you have to just put this one to the system. Okay, then ChemDraw will be enabled for you. So first, we'll go to the PubChem database. So PubChem is one of the largest database that contain almost 121 million compound and 335 million substance with 295 bioactivities. So here we can explore our knowledge. Here we can explore our compounds, all physicochemical properties and other properties, as well as their chemical structures and their spectra, etc. We can search the compound by using their name, by using their PubChem ID, by using their canonical smileys. Okay, so first let's learn with the name. Okay, because if I am talking about any chemical compound ID, then the question will be as that's where we found that ID, right? So if you have the experience of reading any articles or any research journal, that you can find the ID that they will mention the ID number of a compound that's retrieved from the PubChem or from the gene database or from the drug bank or from the ChemDraw or other. Okay, so let's start with the name first. So I'll input the name myrin, which is the synonym. Sorry, it's a name mistake. Okay, so this is the synonym of sometimes butyllinic acid, myrin, or butylic acid, or lipoic acid. Okay, so these are the synonyms, and you can see that the name myrin underlined with a yellow color. So yes, this is the myrin compound, and its chemical ID or CID, which is the PubChem ID, is 64971, and its molecular formula, its molecular weight, IUPAC name is Myris, inchi, injiki, etc. and the date. So let's enter. So here all information we will found here, and the list of content are listed in the right side. And the first title and summary is structure, the name and identifier, chemical and physical properties, spectrum information, okay, related records, chemical vendors, drug and medication information, pharmacology and their biochemistry, use and manufacturing, safety and hazards, associated disorder or disease, literature, patents, and so on. Okay, so first the PubChem CID, and then structure available in two form, 2D and 3D, and we need only the 3D structure for further analysis. Okay, so the availability of 3D-dimensional structure is important here. So we have to first check that is the 3D structure of this compound are available or not. Okay, otherwise we will search this one on another platform. Okay, and then no need to extract this properties from here, because we'll learn to calculate these properties of any compound in our another class. Okay, so we just need the structure. You can assure about the structure by seeing 3D, 2D, and then here is the 3-dimensional structure or 3D structure. You can see you can rotate this in any dimension. Okay, and here the name and identifier, such as their IUPAC name, inchi, inchiki, smileys, their molecular formula. Okay, and other these are the other external link where you also can find this drugs, such as this is the drug bank cited, this is the Campbell ID, and okay. And these are the synonyms of this compound, available synonyms. So, and then from here you just download this coordinator file. So for this one we have to click on this download coordinates and then choose the SDF file format. Okay, and we have to choose the SDF file format, not the JSON or XML or ASNT, only the SDF file format. Okay, we have yes.
Sorry, my net was really slow. You were lagging a bit. Where do you send the link for this?
So for this website,
Yeah.
just you can just search them as PubChem, and just this is the PubChem database. Okay.
Oh, okay. Thank you.
So we just downloaded this structure, especially the 3D conformer, and also at a time we have to generate a spreadsheet or Excel. Okay, or you can keep this information using your Word documents file. I prefer Excel or Word sheet, Google Excel. So just let me open my Excel sheet. Now we'll create some columns here as the name of compounds. Okay, name of compounds, and their CID. So CID means the compound identity, or PubChem ID, or whatever. We can use the ID number, and their smileys here. Okay, smileys. So these three information are very important. We'll need this information in our next class. Okay, when we will calculate the physicochemical properties of these compounds in our pharmacokinetics classes. So that's why we collect this information at a time. So just you have to do the same thing, like have to just put the name, okay, and then the compound ID is 64971, and just from here the name and identifier, collect the smiley. Okay, just collect the smiley from here. So this information are mandatory for you. Okay, if you are if you want to do some organized work, then you have to be organized from the first of your research. Otherwise this work will not work fully at the end of your research. So then so this is all about from the PubChem database. Actually we also collect some more information from PubChem database, and I will back again in this database after some times. Okay, so from this database, how to retrieve and how to search any compound here, and how to collect their information and the structure availability from this website. We just learned it, and now we'll go to another website which name is the drug bank. Okay, so here is the drug bank. Let me explain the difference between these two websites or two databases. Okay, let me okay, search our knowledge base. Okay, 5000 K plus drugs and the drug product. Especially in drug bank, we can found that drug only that are approved by the FDA or other approval association, or that compounds that are on the current research in preclinical trial or clinical trial. Okay, not any compound that are not considered for the preclinical or clinical or the FDA approval. Okay, so only the compounds which are approved by any association or which are considered for the clinical or preclinical trial. So if we found any compound, then it should be under review for the clinical and preclinical. So let's start with the name, naringenin, and search. So you can see that the drug bank ID is here, and the modality is the small molecule. So US approval is no, and other approval no. There is no patent and indicated. So clinical trial is not yet started for any phase, might be in preclinical trial. Okay, so here you will find the name of naringenin and their section number. This is the structure. Okay, is the structure of naringenin, and the 3D structure you can found here. Okay, so let me refresh the page again. Okay, here is the 3D structure of naringenin. And that is the download option. Okay, in drop down menu. So is the option for mole, SDF, 3D SDF, PDB, smileys, and int. So these are the text documents file, and also these are the text documents file. The mole file, the SDF and 3D SDF. What is the difference between this SDF and 3D SDF? So first one is the 2D SDF file, and second one is the 3D, as mentioned that it is 3D SDF file. So if we click on 3D SDF by clicking our left button, okay, then let's see, it just converted to another page in a text format, okay, not in a SDF file format that we see in the PubChem database. So if you want to like that compounds file, then we have to just put our cursor in the 3D SDF file format, then click on our right button, and then save link as, okay, then save link as. So save this link as it is, and then just save it. You can download the PDB, okay. So for PDB one, save link as .pdb, okay, .pdb, okay. So let me first collect this all compounds from every database, and then we will open this structure and we will discuss more about this structure and their complexity, and the difference between 2D and 3D structure, and why we consider only the 3D structure, and why we consider only the PDB or the SDF file, or okay. So in case of drug bank, we can
Can you show how to download the 3D SDF again?
so sorry,
How do you download the 3D SDF again? What do you do after you get the screen with the black screen, all the white numbers and letters?
okay, just wait and see, okay. And if you have any question, then please, we will consider your question after completing the class.
Okay, thank you.
Just note down it, okay. As well as you can see the information about these compounds, its pharmacology, its interaction, categories, chemical identifier, references, and also you can find about the targets that are already suggested against this compound. So here it shows about 10 targets, okay. You can see the ASTH type transcriptional regulator, okay, and also all the 10 compounds are here, and about their literatures, the references, okay. And so that's the pharmacology. So as it is not in clinical trials, so there is no indication are available that which purpose it is used, and the pharmacodynamics are not available. So only the mechanism of action, as it mentioned that about targets, so mechanism of action are available here. So it is an inhibitor or an agonist or antagonist, okay. So there is some for this antagonist, and other in case of this inhibitor. And also their pharmacokinetics properties are not available. So this compound could be a good target if, as it is not approved, or not there no identity about the pharmacology, pharmacodynamics and pharmacokinetics, okay. Now all about the drug bank database. So if we want to download any compounds from here, you can, but I just introduce this one to you just because of an alternative option. Okay, but my recommendation my first recommendation is about the PubChem, and in case of second one is the Gene database. If we search about the Gene database in Google, then this is the Gene database. Okay, gene.docking.org. So docking.org, Gene database. So Gene database is considered as the largest database for compounds. Okay. Sometimes it takes too many time due to the internal server issue. Okay, so we're trying to collect individual compounds from Gene database or the PubChem or drug bank, right? Now let's try to collect some analog compounds. Analogous are their similarly structured compound, or you can say the derivatives. Though the derivative and analogs are different in case of chemistry. So just search the name naringenin or anything you want. So you can see that naringenin is here, the best match. Okay, this is the best match. So this is the best match, and also this name is separated, and this is the actual naringenin compound. Okay, and another 131 results are showing here. So these are the match name or the name where the naringenin is present. Okay, and their structure are quite similar, and they are from single structured compound like this one. Okay, this ring, from this ring. So now we will try to search the similar structure. Okay, so if we want to collect this 131, these are the most similar compounds and best match, or we can search them by using this similar structure search option. Okay, so if we use this similar search structure option, then we will found a more compound related to this structure. Okay, more compound related to this structure that are converted from this one, or that could be extended by using this compound structure. So could be modified there, any functional group or any bond. Okay, so it shows 1000 result incomplete. Now we can download these compounds and their information. So here is the download option. Okay, the summary, you can download the summary as CSV file. Okay, as JSON and XML. The chemical structure record 2D and 3D. Okay, so I prefer 3D, and you can also use 2D. So 3D, you can use it as zip file, and the chemical structure image is small or large option. So first, let's reduce the number by applying some threshold value. Okay, so just click on this filters, and the molecular weight, limit the molecular weight. So we can see here the maximum molecular weights are 750. we can we just limit at 500, and we can see the number is reduced to 989. Then the number of rotatable bond counts here are 14. We will limit it in 10. Then also this number is should be below 30. Okay, so you can use the 30. Okay, why this number I'm choosing, I will not explain this criteria in this session. Okay, because for this reason and the lecture are different, and we will discuss in our pharmacokinetics class. I'm just showing you to filter the data from the PubChem database only. So the number of hydrogen bond donor count here is maximum 7, so it should be less than 5. Okay, and the number of hydrogen bond acceptor should be 10, less than 10 or equal to 10. Okay, it's within the range, and the polar surface area also are within the range. It should be less than or equal to 140 angstrom. The standard range or the maximum range is 140 angstrom from the okay. And the log p value or the lipophilicity index should be less than 5 or sometimes 4.5. okay, so just 5. okay, we set all the parameters here: the molecular weight, rotatable bond, heavy atoms, hydrogen bond acceptor and donor, polar surface area, and log p value. And the number now are 717 compounds are there. So we can now choose this complex, this compound for further analysis, or we can choose any number based on our study. So this is the way in which you can download your analogous compound or your derivatives compounds of any from your any compound structure from the PubChem database, and these are the ready to run your docking and simulation results. Okay, maybe the server for Ching is not available, service unavailable. Yes, so this is almost same as PubChem. Okay, so as it is now unavailable, I will discuss on it later. Now let's talk about another source of compound. As we mentioned in our last session, that with the couple, sure.
Are we doing all this to find our target?
No, this on this class is only belongs for the compounds only. Okay.
Okay, thank you.
Okay. So as we mentioned in our last class that we can choose the compounds from two sources. One could be a chemically synthesized compound, or chemical compound that could be synthesized in your lab or could be synthesized from other minerals. Okay, and another source is from nature, or especially the plant. Okay, so plant-based compound. So that's why now we'll know about database from where we can download easily the compounds that are derived from any medicinal plant. Okay, so this compound is this database is specially designed from the Indian medicinal plant. That's why this is named as IMPACT. Okay, IMPACT. So if you search as IMPACT on Google, then you can see the Indian medicinal plant and phytochemistry. Okay, so impact for website. This website is also down today. It's nice. I see. Okay. There's nothing to do. I'm trying to enter this website from last 2 days, and still it is now under maintenance. So okay, we will discuss on impact and zinc later when it will okay for us. So now the question is, if we do not found any compound in this database as the 3D structure, then what we should do? Okay, for example, if you can see that only 2D is available here, 3D is missing. Okay, then don't worry, we will now try to convert this 2D to 3D structure, and we'll learn how to draw any compound using ChemDraw database and how to convert this 2D compound into 3D. Okay, so for that, let's download the compound's 2D structure. Okay, let me show you the difference between 2D and 3D. Both are the SDF format. You can as you can see that this structure is 2D and this is the 3D, but both are the form as SDF, and this is also in PDB. Okay, then what are the actual difference? Right, let me show you. Just let me open my folder. I will get back to this one. Okay, we're good to go now. So here we download the 3D structure, as it named as conformal 3D, and the 2D structure is named as structure 2D. Okay, and this is the drug bank as PDB. Okay, so we can choose the extension of TXT in SDF easily by writing this on SDF. Okay, so now just open the Avogadro file, Avogadro software. Open it. Okay. Okay. Now let it open here. Okay, so when I'm open in this interface, the compounds are okay, and this could be rotated. But when you open the 2D structure in Avogadro, just notice here, and notification is arrived here about the warning that this file does not contain any 3D coordinates. This file does not contain any 3D coordinates. Okay, so this tools is one from your permission that do you want to allow Avogadro to build a rough geometry. Okay, this will produce a rough geometry for you, and this will be okay. But this will not work. Okay, after completing the molecular docking study, this compound will break down in several fragments. Okay, so this will be fragmented after molecular docking, and your binding will not work properly with the target receptor protein. Okay, So for this one we have to do another thing, like just open it on Discovery BioVia Discovery Studio. Okay, so for first time, if you are using the BioVia Discovery first time and open any compounds, okay, then this type of interface will show. Don't worry, just go to the view, okay, enable the graphics, okay, enable the graphics. So here is the graphics, and now again go to the view and enable the hierarchy. Okay, so here is the compound ID and the compound name or identity number is here. Again, now you have to select this one, okay, just select this one, go to the chemistry, hydrogen, add hydrogen, okay, again go to the chemistry, hydrogen, add polar, okay. Now go to the file, then save as PDB or SDF as you want, or you can use it as mol2 file format or other present. So I chose the SDF file format, so the extension is SDF. So I'll just rename it as the 3D structure. Now let me open it. So file, then open from this one, 7 batch, then +2, and the 3D compound. Okay, now the warning is gone, right? So this is the one method, and sometimes this is the cheapest method, okay, because the error in case of docking could be produced if we use this method sometimes. So now we will try to draw this compound using ChemDraw, okay, to reduce the possibility of any error during the study. So let's open up the ChemDraw. So is my ChemDraw. Okay, so your ChemDraw will not appear as my one, okay, because for this one, view, the crosshair and the rulers, okay. So if you want like this, just enable the crosshair and enable the rulers. They are the ruler for align properly the reaction stoichiometry. Okay, let me introduce you to this software. So this could be disappear in case of your interface, okay. So to bring this periodic table here, you just go to the view, okay, and show periodic table window. So then this periodic table window will be appeared here. Now this is our working toolbar. These are the selection tool, okay. These are the structure perspective tool, okay. Eraser, the text, a single bond, double bond, and if you click it, then you can see that there are several bond types, okay. There are several bond types, okay. And the other bond types, dot, with, hash, okay. And you can insert the charge, anion, cation, and their transformation, okay. and the brackets are there. And the shape, different type of shape you can choose from this window. And these are the orbital geometry and the arrow. Okay, this is the pen tool, and here is the structure of benzene or other painting, other hexagonal or conformation, pentagonal conformation, octagonal or others. And we can see that the chairboard conformation also present here. Okay, let me draw a rough geometry of any compound. Okay, so let's start with the benzene, right? This is the benzene. We all know this one. So this is the benzene structure. Okay, Kekul's benzene structure. And now, okay, if I want to download this structure, so for this one, okay, so here is the benzene ring. We consider as this one. Okay, now select the single bond. Okay, single solid bond, insert here. Okay, for this one you have to notice carefully that if the box is appear where you want to insert any molecule or any bonds, then it will work perfectly, otherwise not. So okay, this is here. And now, sorry about it. So this type of, and here is the oxygen. So for oxygen, just choose the oxygen from here and insert it. And now here is the hydrogen. So hydrogen, and from, okay, you also can insert the molecules symbol using your keyboards. Okay, for example, just you want to press the nitrogen here. Just press nitrogen, and just simply press the N of your from your keyboard, or the oxygen or O from your keyboard. Okay, for carbon you can choose the C. Okay, for sulfur you can choose the S. Okay, so let me choose hydrogen. Again, the single bond. Okay, this one and this. Okay, so hydrogen and this is the oxygen. This is the hydroxyl bond. Now again, ring here, hexagonal ring. So yes, and here is our oxygen and a double bond. Okay, and oxygen. Now again a single bond, and our benzene ring is here. So just down. Now complete the skeleton. The oxygen, and yes, this is also the hydroxyl bond. going on, and okay. So just click on the selection button and it will be selected. So this is our final structure, okay, of our desired molecule. Now if we draw any compounds from the outside of database that are missing here, the information are missing in the website, you also can produce, or you also can generate any compound that are novel, okay, whose information will not available in the database. For example, its name, for example, its smiley and the IUPAC name. So then as I mentioned that we need smiles of this compound for our further work. So if we draw any compound in ChemDraw, then how could we collect the smiley, right? So also there is a possibility to collect a smiley from ChemDraw. So for that reason we have to select our entire complex. So we just select our entire complex and go to the edit, okay. Now copy as, then you can copy as the smiley, okay, SLN, inchi key, and mole text mole, okay. So just copy as mole smiley format and paste it here. So this is the smiley. Okay, so this is the smiley. Now it's time for name, right? So again select, go to the structure, convert structure to name, okay, convert structure to name, and this is the name of your compound. Okay. Again, there are some options for coloring like this one, okay. In case of elements, nitrogens are blue, oxygen are red, and other molecules has other color pattern like this one, okay. Here you can see the blue, black for carbon, and the iron has its own color, sodium has its own color for copper and gold, and sulfur, phosphorus, okay, chlorine, bromine. So just select and go to the color and color by element, okay. Then this will look like, as you can see that in the periodic table here. So view, okay, here is the periodic table. So the hydrogen has no color, but if you want, then you can choose the color other and then choose it, okay. Then the color, choose it. Okay, so now it's okay, it's okay. It's time to save this file, okay. Just go to file, save this PC into shift here, 0725 batch, and now engine, okay. So the original document type file of ChemDraw is CDX, Now we have to convert this CDX file to mol2, SDF, PDB. Okay, so go to the file, save as, and save as type. Just click on save as type. You can find several types of for saving this. Okay, like GIF image file. Okay, and it's PNG file, and also here you will find the mol, SDF. This one is SDF, or this one is SDF. So MDL SD file, MDL SDF file. This one, okay, SDF file, and here let me save it as it is. Yeah, and okay. Okay, so just go to the folder. Sheet, the naranj is here. So Control G and Control Ace. If you want to enable the graphics, press Control G, and for hierarchy, Control Ace. You can also find the command here, Control G and Control H. So this is the naranj. Okay. Now let's check whether it is 2D or 3D. Okay. So open and file, open the file from our cat2 and the naranj SDF file. Okay, it shows that this file does not contain any 3D coordinates. So this is not converted to 3D appropriately. This is only the SDF file format, not 3D file. Okay. Okay. So what should I do now? The main question is just select this one, and right from your right button, copy it, go to the Discovery Studio, go to File, New, and Molecule window. Okay, here you will find a molecule window, then just paste it. We just paste it and it is selected. Now go to the chemistry, hydrogen, add. So hydrogen are added, and then go to chemistry again, then hydrogen, add polar. Now save it as SDF, or you can save it as mol2, or you can save it as PDB. So let me save it as SDF. Again naranj. So now the 3D structure. Okay, now the 3D structure. So let's open the 3D. So 3D is here, and okay. So these are the total methods for converting your SDF file to especially the 2D SDF to 3D SDF. Okay. Now why we choose this SDF file format? Why not PDB, or why not other like mol2? We also can choose the mol2 file format and PDB, and especially the PDB and SDF is the perfect file type for the molecular docking. Okay, so as we mentioned that we are working on some special tools for molecular docking, and every tools has its recommended file type. Okay, that the tools can read. So this file are the recommended file types for molecular docking, especially the SDF and PDB. That's why we choose the SDF file format for ligand or for any compound's 3D structure. Okay, for this one, if this compound are noble, then you also can calculate the NMRP using the ChemDraw. Okay, theoretical value will be appear here. So just go to structure. Okay, predict proton NMR and carbon 13 NMR. Okay, so if you predict the proton NMR here, then you will see the ppm or shift value. Okay, they are the shift value, and these are the peaks appear for different types of proton, only for the proton. Okay, these two are the proton, and this is the protons. Okay. So if you can read the shifting and shielding, deshielding, and if you have the knowledge of shielding and deshielding, why these compounds are shielded and why these compounds are not deshielded, and why these peaks are appear in left hand side and why these are in the right hand side, then you can predict the structure easily. Okay, now here the all value appeared here, and similarly we can predict the carbon 30 NMR. Okay, these are the carbon. We calculate the NMR only for the proton, that are associated with highly electronegative oxygen atom only. Okay, that's why these all are deshielded. Sorry, shielded. And now just go to structure, and let me pause this one. Okay, no. So now again, if we go to the structure and predict the carbon 30 NMR, then the carbon 30 NMR will be here. So okay, so as you save it as CDX file, so we can edit this file type anytime. So nothing is there more. I think it's okay. We shows the all types, so we can expand the label types, contact, and we also calculate the atom properties and bond properties. Okay. So in ChemDraw there is a special service. I can show you. Okay, let me collect IUPAC name. Okay, let's say this is the IUPAC name of this compound. So we can easily generate any compound's structure using ChemDraw within a second. Okay, within a second or within a millisecond. Just go to structure, convert name to structure. If the name is available in their database, this will appear within a second. Okay, so just paste your name here and click on OK. So your compound is ready. Okay, so this is your compound. Okay, so I think it's okay. It could be better if we can see these two database, but this website is down, and also the gene database. Let me try again. Let me show you the website interface. So I'm starting from the first. So we search the gene database. Okay, gene database is the search option, and these are the version gene 12, 20, and okay, we can type 20, 12, or 50, or the natural product catalogs. Okay, so just put the SARS as the main database website is blocked. Okay, that's why we are trying with Jing 12, and the updated version 20 is still now browsing, and I think so this version is under maintenance. So these are the natural product catalogs. Okay, so in search box we can paste the name Narinjani. Okay, and let's search the Narinjani. So there is the two products, Narinjani, and these two are the conformer of each other. So just click on this one. It's the Narinjani. The name is Narinjani, and the other structure and their properties are available here. Okay, so from this menu you can download the mol2 file, sdf smileys. Okay, so just click on sdf, download sdf file. 1 minute please. Okay, they're directing this link to the Jing 11, and this is not working properly. So let's wait for, I think it's better to discuss this one later. So thank you everyone for your patience, and thank you for joining today's session. So if you have any question, then you can ask me here. I'm stopping the recording session.